Today we're looking at this quiet but absolutely furious scramble that's happening in labs and forests all over the world. It's a race against time for biologists.
It really is. It's this desperate fight to name every species on Earth.
And if the trends hold, it's a race we are losing. I mean, losing dramatically.
And that is the staggering, inescapable reality. We know Earth is just teeming with life we haven't even met yet, let alone cataloged.
Right.
And with climate change, habitat loss, we're losing them faster than we can find them. It's a huge challenge for science, trying to document all this biodiversity before whole ecosystems just, vanish.
So we've taken a deep dive into the sources you sent over, and our focus today is on this decades-old struggle over how we even categorize life itself. And we're not talking about, lions and tigers. We are using the hyperdiverse and, frankly, mostly unknown world of tiny wasps as our test case.
Specifically, the superfamily Chalcidoidea. They're the perfect subject because they really embody the core problem, which is something called dark taxa.
Dark taxa. Let's define that.
These are groups, usually tiny invertebrates, flies, midges, and these parasitoid wasps we're talking about, where there are just immense gaps in our knowledge. We don't know their true diversity, where they live, what they do.
And the Hymenoptera order, the one wasps belong to, is estimated to be the single most diverse insect order on the planet.
Exactly. And it's because of the richness of these parasitoids. But ironically, they're the ones we know the least about.
So they're everywhere, they're microscopic, and they're incredibly hard to classify the old-fashioned way. And that difficulty leads us right into the tension we're unpacking today. This conflict between traditional morphology, what these things look like under a microscope, and the new molecular tech like DNA sequencing.
And then finally, we can look at the really innovative AI-driven solutions that just might be what we need to finally catalog the millions of invisible life forms around us. Life that we're losing simply because we don't even have a name for it yet.
Okay, so let's start at the foundation of this whole crisis. The idea of cryptic species. For anyone listening who thinks taxonomy is still mostly about, looking at things, what exactly is a cryptic species?
A cryptic species is an organism, or a group of them, where the individuals look identical. I mean, even to a world-renowned expert, they're visually indistinguishable.
Okay.
But when you look at their genetics, they are demonstrably distinct species. They don't interbreed. Their DNA is different, but their external appearance has just sort of stayed the same.
The sources have this incredible example from Australia that just shows the scale of this. The Australian minnow or the mountain galaxias, if you were catching these fish, experts would have said, what, one or two species?
Yes, exactly. And that was the assumption for decades. But when researchers did comprehensive genetic testing, going way beyond just how they look, they found something, well, astonishing.
What was that?
That single complex of what they thought was one or two fish. It was actually 15 just reproductively isolated species.
15.
15, all hiding under one name.
Wow, that just completely collapses any traditional conservation assessment, doesn't it? You go from thinking you have one common, widespread fish
to maybe 15 highly isolated, rare species, many of which could now be facing extinction. It just shifts the entire conservation priority overnight.
And that's the key, isn't it? The consequence.
It is, and this isn't a rare thing. It's rampant.
Yeah.
Especially in places like agriculture, where we really need to be precise. Take the sweet potato whitefly.
A major pest.
One of the world's most damaging. For years, we thought it was one species. Genetics revealed it's a complex of at least 28 visually identical species.
28. And I assume they all react differently to pesticides.
Exactly. And have different host preferences. So a pesticide that works on the whitefly in one field might completely fail in the next, and now you know why.
It seems like the complexity just grows the smaller the organism gets.
It does. And the problem is particularly acute with the insects we're focusing on. Parasitoids. A lot of the wasp species we study were historically labeled as generalists. They attack lots of different hosts. But when you apply genetic testing, those generalists frequently break down into complexes of cryptic species, and each new species is actually a highly tuned specialist that only attacks one or two hosts.
Which puts the person trying to identify them, the taxonomist, in a really tough spot. I mean, the sources mention that extreme environments like caves often lead to species that look the same. If you can't tell them apart, your count of biodiversity is just fundamentally wrong.
It's biased towards undercounting, massively, which is why we have to move beyond just counting species names. And this brings us to a slightly broader but really important ecological concept, dark diversity.
Okay, let's dig into this. If cryptic diversity is about species we missed because they look the same, what is dark diversity?
So dark diversity is an ecological measure. It's the set of species that are absent from a specific place, say a patch of forest, but which could potentially live there because the environmental conditions are right.
So it's like the missing potential of an ecosystem.
That's a great way to put it, yes. If you have a forest plot with the perfect soil, moisture, and light for a specific rare orchid, but that orchid isn't there, that orchid is part of the dark diversity for that plot.
And that tells you what?
It helps us understand why a community is the way it is. Is it missing species because the environment is too harsh? Or is it because those species just haven't managed to get there yet? It's about dispersal.
But the sources make a really interesting distinction here, moving from just counting to, well, to function. They compare taxonomic dark diversity with functional dark diversity. What's the difference?
Right. So taxonomic dark diversity just counts the number of potential species names that are missing. Functional dark diversity goes deeper. It looks at the missing ecological functions or traits.
Like what kind of traits?
Dispersal methods, feeding habits, things like that. It assesses what jobs are missing from the community that should be there.
Okay, so let's look at that study comparing this in old versus new forests. They looked at plants and spiders. What was the hypothesis?
The researchers basically wanted to know if counting names gives you the same answer as analyzing roles. Their core idea was that functional dark diversity would show you something different, something the simple species list couldn't.
And what did they find? When they compared the ancient old-growth forests with the newer, re-established ones?
The patterns were really different. For plants, the taxonomic dark diversity, the number of missing species, wasn't that different between the old and new sites.
Okay.
But for spiders, the taxonomic dark diversity was much higher in the new forests. It suggested that these newer habitats were struggling to attract their potential spider residents.
But the real aha moment, as the outline calls it, came from the functional assessment.
Yes. This is where it got really interesting. When they analyzed the functional dark diversity, they found patterns that were totally different from the taxonomic results. For instance, with the plants, while the number of missing species was the same, the types of missing species, the ones with certain traits, were vastly different.
And what was the big function they found was missing in the newer forests?
Dispersal limitation. The taxonomic approach didn't really show it, but the functional approach revealed it was a huge factor. In these new forests, even decades later, species that needed specific, slow ways of getting around were disproportionately absent.
Like plants with heavy seeds or insects that can't fly very well.
Exactly. So just knowing that, say, plant X is missing doesn't tell you a whole lot. But knowing that all the plant species that rely on a specific animal to eat their seeds and move them around are missing.
That tells you that your disperser community, the animals, might be compromised. The ecosystem isn't complete.
Precisely. Functional dark diversity is a much better tool for understanding ecosystem health. But to understand the functions, you first have to accurately identify the species that perform them, which brings us right back to the central problem of taxonomy.
It's a circle. You can't know what functions are missing until you know what species exist, and you can't identify all the species without radical new tools. And this tension brings us right into the human side of science, this classic long-running conflict between the lumpers and the splitters.
Oh, this is as old as the field itself. It's about how you deal with variation. Lumpers, they tend to prioritize similarities. They want big, broad, inclusive categories.
Yeah, right.
Splitters are the opposite. They emphasize the differences, making very precise, narrow definitions. And that inevitably leads to creating a lot of new species categories.
We see this in all sorts of fields, but in biology, the stakes are very real.
And this argument involved the absolute giants of the field. Joseph Dalton Hooker, a famous botanist and a contemporary of Darwin, was a fierce lumper. He wanted to find these broad philosophical laws for botany, these global patterns.
And how did he see the splitters?
As administrative roadblocks, basically. He argued they just created tons of redundant synonyms, multiple names for what he thought was the same thing, and all that noise made it harder to see the big picture he was after.
So for him, it was just getting bogged down in detail. But the sources show this is a cycle, and new tech often forces a reevaluation, like the example of the common gallinule and common moorhen.
That is a perfect modern case. It really shows how a decision to lump based on limited data can be overturned. So in 1923, they were lumped into one species. They look almost identical. Their breeding ranges overlap. It made sense at the time.
They were just subspecies.
Right, until 2011. Nearly a century later, new evidence comes in. Differences in their calls, subtle but measurable differences in their bills, and critically, mitochondrial DNA.
And the DNA sealed it.
It proved they were distinct, reproductively isolated species. So the split was confirmed.
And what's so fascinating is how molecular data, like DNA, almost always seems to tip the scales in favor of the splitters, especially with insects.
Oh, absolutely. That's where you see its real power. There was a case with a skipper butterfly in the Neotropics, Astraptes fulgurator. Based on how it looked, everyone assumed it was one single widespread species.
But the DNA told a different story.
A very different story. The molecular data showed deep, significant splits. That one butterfly turned out to be 10 distinct species.
10 species, all looking the same to us, but genetically they never interbreed.
It's undeniable proof that morphology just couldn't see. And this, of course, led to this huge wave of optimism around DNA barcoding. It felt like the silver bullet for the dark taxa problem.
But as always, it's more complicated. The sources quickly get into the limitations of relying on just a single gene for this.
This is such a critical point. DNA barcoding usually uses one specific mitochondrial gene called COI. It's fast, it's cheap, and it works great for many groups. But for hyperdiverse, ancient groups like our wasps, it runs into some really tricky biological problems.
Okay, let's break down those pitfalls. What are the biological issues that make that COI gene signal unreliable?
Okay, so you have three main problems. The first one is something called NUMTs.
NUMTs.
Right, it stands for non-functional mitochondrial DNA. These are basically dead copies of mitochondrial genes that have, over millions of years, gotten stuck in the cell's main nucleus.
And why is that a problem when you're sequencing?
Because you might accidentally sequence one of these dead copies from the nucleus instead of the real, living one from the mitochondria. And those nuclear copies evolve much, much slower. So you might think two species are really closely related, or even the same, when they're not. It can lead to false lumping.
Okay, that makes sense. And what were the other two issues?
Uneven race of evolution across different groups and something called genetic introgression.
Introgression.
That's just gene flow between two different species. Sometimes, even if two species are distinct, they can hybridize and swap certain genes. If the COI gene gets swapped, it blurs the signal. It makes them look like one species at that one spot in the genome, even if the rest of their genomes are totally different.
So the biological signal itself can be messy. And if the signal is messy, I'm guessing the computer programs trying to read that signal must also have problems.
Oh, this is the biggest headache. It's called algorithm incongruence. You have all these different automated species sorting algorithms. They have names like ABGD, GMYC, PTP, and they all operate on different principles.
What's the main difference in how they work?
Well, think of a method like ABGD as being distance-based. It just looks at the genetic difference between two sequences and says, if it's more than, say, 3% different, they're two species. It's a simple threshold.
Okay.
But others, like GMYC or PTP, are tree-based. They build an evolutionary tree from the data and then look for points on the tree where the branching pattern suddenly changes. They're trying to find the statistical point where populations stop mixing and start evolving into separate species.
So one is just measuring distance, the other is trying to infer a whole evolutionary story. I can see why they'd give you different answers.
And they do. There was a study on the Parasitoid Wasp family, another one of these tiny, hard-to-tell-apart groups. They ran the same DNA data set through these different methods.
And what was the result?
The number of species they found ranged from 55 with the most conservative method all the way up to 123 with another.
That's more than double.
It is. And if the algorithms can't agree if you have 55 species or 123, it tells you that the molecular result on its own is just a hypothesis. It desperately needs to be checked with other data, like morphology.
And for the really tiny things, like the Malaise wasps, the fairy flies, that's almost impossible. The sources said their DNA reference databases were basically useless.
Less than 1% correct species matches. It just shows you how deep the taxonomic neglect is for some of these groups.
So when we try to put these tiny insects on the big evolutionary tree, it all gets chaotic.
Chaos is the right word, especially at the higher levels. So molecular studies do strongly support that the whole Chalcidoidea superfamily is one group, which is good. And it confirms that the Maiameridae, those fairy flies, are the oldest branch.
But once you go deeper into the family tree, it all falls apart.
Precisely. The relationships among the families, what we call the backbone of the tree, are super variable and poorly supported. The results are often described as a broad polytomy.
A polytomy. Can you break that down for us?
Yeah, a polytomy on an evolutionary tree is a node where 3 or more branches come off at the same time. It looks like an asterisk. It means the data isn't good enough to figure out the exact order of branching.
So it's not that they aren't related, it's that they all evolved and diversified so quickly, so long ago, that the genetic signal of who came first is just lost to time.
Exactly. And that deep uncertainty just highlights how hard it is to classify these groups. The molecules show us some traditional families hold up, like the Eulophidae, but others, like the Chalcididae, seem to be completely unsupported by the genetic data. It forces us to admit our visually based system has some serious flaws.
So, okay, the molecular data is showing the limits of morphology, but the molecular data itself is so complex it creates its own problems. We need a way to combine their strengths.
We do. And that's where the field is now.
Which brings us to what's considered the gold standard today, integrative taxonomy, or IT.
Right. IT is really the recognition that we can't rely on just one type of evidence anymore. You have to combine multiple independent lines of evidence: morphology, DNA, ecology, behavior, geography. You put it all together.
And the idea is that if 3 different types of data all point to the same species boundary, you can be pretty confident that it's a real boundary.
That's the idea. It's incredibly rigorous. But
it's slow.
it's way too slow. If you have 1,000 wasp specimens and you have to collect 3 different kinds of data for every single one, you'll never catch up. We're losing the race.
We need the quality of IT, but at an industrial scale.
Which is why this new protocol was developed. Large-Scale Integrative Taxonomy, or LIT.
Yeah.
And the genius of LIT is that it completely reverses the traditional workflow.
How so?
Traditionally, an expert would sort everything by how it looks first. That's the bottleneck, and it's where you miss all the cryptic species. LIT flips it.
So what's step 1?
Step 1 is cheap, fast, automated data for all specimens. Usually that means next-generation DNA sequencing. You barcode everything first and let the computer sort it all into preliminary molecular groups, or MOTUs.
Okay, so you get a first draft from the DNA. Then what?
Then step 2 is selective validation. You use data-driven rules to flag only the weak hypotheses. These are the molecular clusters that look suspicious, maybe the genetic distance is really low, or different algorithms disagree on it. Those are the clusters that are most likely to be hiding multiple species that have been incorrectly lumped.
So the computer does the bulk sorting and then flags the problem areas. And the human expert's time is spent only on those flagged cases.
Exactly. Instead of spending 90% of their time confirming what's already obvious, they spend 100% of their time investigating the 5 or 10% of cases that are likely to be new discoveries.
That's a massive efficiency gain.
It is. The study on foreign flies showed just how effective it is. They found they only needed to apply the second, more expensive data type, in their case detailed morphology, to a tiny fraction of the specimens, about 5% of the total collection. That worked out to be about 2.5 specimens per molecular species. By focusing the experts' time just on those high-probability cases, they accelerated discovery by an order of magnitude.
It totally changes the job of a taxonomist. Their skill is still essential, but it's applied in a much more targeted way. And we see this in that Zonocminia wasp case study.
Yes, the Zonocminia wasps. Based on morphology alone, experts had lumped all the specimens that looked like D. brevifunis into one single species. They saw variation, but they just assumed it was normal variation within a species.
Classic lumping.
Very classic. But then they applied the LIT workflow.
And the molecules told a different story.
The molecular algorithms, ABGD and GMYC, split that one species into 3 distinct ones. And here's the crucial part. Once the molecular data told them there were 3 groups, the experts went back and looked at the morphology again.
And then they could see the differences.
Then they could find the subtle, previously dismissed differences that correlated perfectly with the 3 genetic groups.
So morphology was still needed for the final confirmation, but only after the DNA told the human eye where to look.
That's the essence of LIT. It proves morphology isn't dead, it's just not the right tool for the initial sorting of these dark taxas.
And now we're bringing AI into this workflow. Tell us about the Morphology Molecule Network, or MMNet.
MMNet is the next leap forward. It's a deep learning model, a convolutional neural network that's trained to automatically integrate both the images of the specimen and its DNA sequence data to identify it.
So we're training a machine to look at a picture of a wasp, read its DNA, and tell us its name. How well does it actually work?
The accuracy is really impressive. It was getting 98.1% for beetles, 98.8% for butterflies, 96.3% for fish. I mean, really high scores, even with closely related species.
Did the model favor one type of data over the other, the image or the gene?
The analysis showed what you'd expect from integrative taxonomy. Both were important. The model was most accurate when it used both. But the genetic data did contribute slightly more to the overall performance score.
which suggests the molecular signal often holds the deeper truth.
For classification, yes. But the real value of something like MMNet might be in finding new species.
How does it do that? If it's only trained on things we already know, how can it find something new?
Through a confidence score. When it identifies a sample, it doesn't just give you a name, it gives you a probability score, like, I'm 99% confident this is species A.
Okay.
So the researchers set a threshold. If a new sample comes in and the AI's confidence score is below that threshold, it gets flagged. The machine is basically saying, I'm not sure what this is. Based on everything I've learned, it doesn't fit well into any known category.
So that low confidence flag is an automated signal to the human expert that says, hey, look at this one. This one might be new.
It is. It's an immediate filter. It lets us process the massive number of specimens we need to, and it points the experts straight to the most interesting ones. It's a game changer for tackling dark taxa.
Okay, so we've laid out the problem and the high-tech solutions. Now let's talk about the stakes. Why does this academic fight over tiny wasps actually matter for, the real world, for agriculture, for human welfare?
It matters because of confirmation bias. In pest control, we assume we know what we're dealing with. We assume we understand the target organism. Cryptic species completely shatter that assumption.
Because different but identical looking species will react differently.
They'll react differently to pesticides. They'll be attacked by different biocontrol agents. They'll thrive in different temperatures. If you misidentify the target, your multi-million-dollar control program could fail for no apparent reason. Or worse, you could cause unintended environmental damage.
The sources gave a really clear example of this with a biological control program for water hyacinth.
This case is so important because it gets at thermal physiology. Water hyacinth is this horribly invasive aquatic plant. So to control it, scientists released an insect, Tarsus catarinensis. For years, everyone treated it as a single species.
Until the genetics showed it was a complex.
Yes, exactly. Genetics revealed two cryptic species, the original one and a new one, E. eichhorniae. They looked identical, but their physiology was critically different.
In what way?
Their response to temperature. The new species, eichhorniae, was actually better at damaging the weed, but only at high temperatures. The original species was less damaging overall, but it worked much better in cooler temperatures.
So if you were trying to control water hyacinth in a cooler climate and you release the heat-loving species,
your program would be a complete failure, or at best, it would be minimally effective. You were deploying the wrong tool for the job, but you didn't even know it.
So the key takeaway is that taxonomy is now about precision ecology. You have to match the right cryptic species to the right environment.
You do. Taxonomic precision directly impacts the success of global agricultural programs. If we want sustainable weed control, we have to be sure we're deploying the right biological agent.
And this need for precision is even more critical when we talk about the tiny parasitoid wasps that are used all over the world in agriculture.
Right. These wasps are our natural allies against pests. But the problem is that a wasp sold as a generalist, one that supposedly attacks many pests, often turns out to be a mix of 5 or 6 highly specialized cryptic species.
So if you release it in a field, only one of those 6 specialists might actually target the pest you have, and the effectiveness just plummets.
Exactly. And then there's the other risk, non-target attacks, like what happened with the gorse pod moth in New Zealand.
This was a cascade of errors, wasn't it?
It was. The moth was released to control invasive gorse. The original population they tested from England was very specific to gorse. But through poor management, they also ended up releasing offspring from an untested population from Portugal. And that one had a much broader host range.
So that was the first failure, releasing an untested population.
It was, but it got worse. The reason it ended up attacking native plants wasn't just because it had a broader appetite. It was because of bad timing asynchrony.
What do you mean?
The moths emerged and were active at a time when the gorse wasn't flowering very much, so their preferred food source wasn't available. And that's what drove them to attack the less preferred but available native plants. It was a combination of taxonomic oversight and a failure of ecological timing.
These examples really show that getting the classification wrong has huge practical and economic costs. And if we zoom out even further, this whole failure to classify life correctly creates this massive systemic bias in our global understanding of biodiversity.
We can actually quantify that bias with huge databases like GBIF. Researchers looked at the data for 24 different classes of organisms, and the imbalance is just staggering.
Which groups get all the attention and which are left in the dark?
A's birds are by far the most overrepresented, almost 99% taxonomic precision in the data. They're heavily researched, they're well documented.
And on the other end?
Insecta, the class that includes our wasps, is the most underrepresented, followed by spiders, snails, and fungi.
So the groups that contain the vast majority of species and ecological functions are the ones we know the least about.
That's it, exactly. Which means most of our big ecological theories are tested and built primarily on the organisms we know best, birds and mammals. And the problem is getting worse.
How is it getting worse?
The studies show that the concentration of data on groups like birds has actually increased the overall taxonomic bias over time. The gap between the known and the unknown is widening.
That's a startling thought. The better we get at studying birds, the further behind we fall on everything else.
And that severely limits our ability to create conservation strategies or ecological principles that apply to all of life, not just the feathered and furry parts.
So this challenge of cataloging the tiny, cryptic, dark taxa, like the Chalcidoidea wasps, it isn't some niche academic problem. It's absolutely central to correcting this huge historical flaw in our knowledge of life on Earth.
It's about making sure our conservation and biocontrol efforts are based on reality, not just wishful thinking.
Okay, so to wrap this up, we started this deep dive seeing this as a race against extinction and found that the biggest hurdle was this old, slow taxonomic workflow trying to deal with just incomprehensible biodiversity.
We went from relying on how things look to the promise of DNA, and then hit all the roadblocks with single gene methods and conflicting algorithms.
But that pressure, that necessity, has pushed us into a new era of integrative accelerated taxonomy. Things like LIT and AI tools like MMNet are completely redefining the process.
They help overcome the sheer numbers problem, focusing that scarce expert time exactly where it's needed most.
And the implication for you, the listener, is clear. This fight over how to name things isn't just for academics. It directly impacts our ability to do conservation, to have successful agriculture, to make sure we deploy the right biocontrol agent in the right place at the right time.
And our whole dive into the Chalcidoidea wasps really highlighted the deepest challenge remaining. Even with massive data sets, over 100 mitochondrial genomes, the deep evolutionary relationships, the backbone of their family tree, are still a total mystery. It's just a big, unresolved polygamy.
Which brings us to our final provocative thought for you to think about. If we struggle this much, with all our best technology, to define the high-level family structure of some of the most diverse groups on Earth, what does that imply about the stability of the entire tree of life as we think we know it?
The true hidden diversity in these dark taxa might, at the end of the day, force us to completely redraw the evolutionary map far above the species level. We might be forced to rewrite the entire history of life on Earth.